With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
In this question, we are asked to select the correct statement regarding the process of transcription in eukaryotes. Right. So the first statement that we have is the strands of the double-stranded DNA which takes part in the process of transcription is called as the coding strand. So if we discuss what the transcription is simply the copying of the genetic information from DNA to RNA. Right. So the first statement that's given to us that the strand of the double-stranded DNA that takes part in the transcription process is known as the coding strand. Well, this is wrong. It is the template strand. Right? It is the template strand that takes part in the process of transcription, which is also known as the non-coding strand. Right, which is also known as the non-coding strand. So we can say that this statement is wrong. Now the next statement is that the enzyme RNApolymerase, it can catalyze polymerization only in one direction, right? And that one direction is 5' to 3'. So this is correct. The RNA polymerase enzyme, right, it basically binds to the promoter region, which is present at the 5' end of the transcription unit, right, and brings about the polymerization, right, only in one direction, that is from 5' to 3', right? And so this statement is correct. Now the third statement is that an unusual nucleotide methyl guanosine triphosphate is added to 5' end of the heterogeneous nuclear RNA during the capping, right? So the primary mRNA transcript is no longer and localized into the nucleus, where it is called as the heterogeneous nuclear RNA or the hnRNA or the pre-mRNA. At the 5' end, right, at the 5' end of the hnRNA, a cap which consists of the which basically consists of the 7-methyl guanosine triphosphate or this 7-mg, right, and a tail of, right, and a tail of poly A, right, and a tail of poly A adenylate residue at the 3' end are added. So these processes are respectively called as the capping, right? These processes are known as the capping and the tailing, right? Capping and the tailing, right? So the cap is chemically modified molecule of guanosine triphosphate, right? So the last statement that's given to us is that during tailing process, the adenylate residue, which is 200 to 300 base pair, that's added to 3' end in a template independent manner. So this is correct, as we have discussed. So that process is known as the tailing, right? So basically the cap is chemically modified molecule of the guanosine triphosphate, that's the GTP, and the primary mRNA is made up of two types of segments, that's the non-coding introns and the coding exons. So the introns, they are removed by a process called RNA splicing, and the exons are joined in a defined order. So we can say that statement 2, 3 and 4 are all over correct, while the statement 1 is incorrect. So these given to us under option 3, that second, third and fourth are correct. So we can say that option 3 is our correct answer.
For class 6 to 12, ITG and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.